I work at Texas A&M University. I've been there now for about 10 years. And the research that I conduct in my lab falls under three general areas. We do some phylogenetics, taxonomy, and also study comparative anatomy of fishes. And in order to do that, we use a number of different techniques that allow us to look at fishes in great detail, ranging from scanning electron microscopy to serial sectioning to clearing and double staining, and more recently CT scanning. Now, the point of my talk today is not to give you step-by-step instructions on how to use all of these different techniques. Each one of these techniques would require a workshop dedicated to it to be able to know the technique well. Instead, what I want to do today is really just to give you some examples of how you can use these different techniques in taxonomic research by using examples from my own projects. In addition to having equipment to look at fishes in different ways, we also are lucky to have access to an aquarium room here at Texas A&M where we can keep live fishes and we can make observations on them, and sometimes we're even lucky that they reproduce, and so we can study early development. And this is really very important for some of the research that we do, especially if we're interested to look at early anatomical stages. Maybe some of the participants in this course are aquarists. It's great to keep fishes at home. That's how I got interested in science myself. When I was 9 years old, my parents allowed me to get an aquarium, and I've had aquariums ever since. You might be able to hear the background noise. That's some of my aquarium filters tinkling in the background. One of the first fishes I ever kept was the cherry barb, which is endemic to Sri Lanka. Beautiful fish. This is a male. The females are not as colorful. But this was the first fish that I kept and bred at home, and it really solidified my interests in fishes. And after I finished secondary school, I went on to do a bachelor's degree in zoology at the University of Glasgow, and I didn't have the opportunity to work with fishes, but I worked with this guy here called Jeff Hancock, who was the curator of entomology, and he showed me the diversity of insects. He allowed me to volunteer in the collection and identify different, mostly flies. He studied Diptera. But I expressed my interest in diversity and identification and classification, and he recommended that I read this book by Ernst Mayr and Peter Ashlock, Principles of Systematic Zoology. And it took me a few weeks, but I read it, and after I finished reading it, I decided that I wanted to be a systematist. And so I went back to Jeff and asked him, how do you become a systematist? And he said, well, that's not very easy to do here in the UK. The only way you can become a systematist is by going to the Natural History Museum in London. And so that's what I did. I moved to London, and I was lucky to get into the master's course there. And you've already heard from Oliver Crimond in this workshop already, and so you know about the treasures there at the Natural History Museum. And of course, they have lots of fishes. They also had an ichthyologist there at the time, Darrell Siebert. Darrell Siebert agreed to take me on as a master's student, and he served as my advisor for my research on the genus Boraris, which are a group of tiny Rasbora-like cyprinids from Southeast Asia. About halfway through my master's research, I bumped into this infamous ichthyologist here. This is Dr. Ralph Britz. Ralph Britz was very nice to me at first. I think the reason for that was because I helped him move a lot of heavy books from his car into his new office when he got to the museum. But he was also very nice to me in other ways. Of course, he actually taught me how to clear and stain specimens. He also taught me how to dissect them, which is, of course, very important if you want to study these specimens. And he also spent a long time teaching me how to make scientific illustrations. And these are some of the first illustrations that I made of the higher palatine arch of some Resbora. And he also told me that it's important in ichthyology that specimens or their parts should always face to the left. And I actually forgot about that. And when I published my first paper, all of my specimens and all of their parts were facing to the right. And so, of course, that was an error on my part. So after getting my master's degree, I moved to the US and I've been there ever since. And I've met up with Ralph Britz numerous times over the years. We've kept in close contact. And in 2007, I actually had the opportunity to take his anatomy course, fish anatomy course that he runs in Brazil periodically. And this is a 3-week course where you spend almost 12 hours a day dissecting fishes and learning all of the details of their anatomy, from the nervous system to the alimentary canal to the musculoskeletal system. It's really a lot of fun. It's also a lot of hard work. This course is produced a number of ichthyologists down in Brazil, and here at the front of the picture, this is George Mattox, who's actually going to be talking in this workshop later today. And if you ever have an opportunity to take a course like this, I highly recommend that you do so. And the course that Ralph teaches has actually had a recent review that came out on the Brazilian Society of Ichthyology bulletin, which talks about the history of the course and how important it is for ichthyology in Brazil. So once you know something about the anatomy of fishes, you really have a much greater appreciation for their diversity. And I'm sure one of the reasons why many of you are taking part in this workshop is because you're just simply impressed by how diverse fishes really are, especially the teleost fishes, which are now numbering somewhere over 35,000 species and counting, because new ones are described weekly. Over the years, I've had the opportunity to work on many different groups of teleosts. And I'm not going to talk about all the projects that I've worked on over the years, but I will focus on some of the groups that I think you might be interested in for different reasons. I'm sure everyone here is familiar with the carps and the minnows and their relatives, and that's a group that I've devoted a lot of my time towards over the years. So I'll start with them, and then I'll move on to the clingfishes, and then I'll finish. So the carps and the minnows, of course, belong to the order Cypriniformes. This is a very large group of freshwater fishes. It's distributed widely across the globe. Its members are found in almost all of the continents, of course, except for Australia and Antarctica. There's over 4,000 described species, but there may be as many as 8,000, and new ones are, of course, described almost weekly. Now, these species are divided across multiple different families, and depending on the author, the number may vary. So we don't have to worry about that. But the family that interested me initially was a family called the Cyrenacus that I'll talk about momentarily. So cypriniforms are characterized by a number of interesting morphological traits. They have a novel ossification in the upper jaw that's called the cynethmoid bone. It's unique to the group, and what this bone allows cypriniforms to do is protrude their upper jaw. The jaws are edentulous, that means that they lack teeth, but they have a very impressive set of pharyngeal teeth at the back of the gill arches. And these teeth can be very important for taxonomy, depending on the group. So all cypriniforms have these characters, and they're very useful synapomorphies that define the group as a monophyletic. So the group of cypriniforms that I was initially interested to work with belong to the family Cyrenacus. This is a relatively small group of cypriniform fishes that was recognized by Indian ichthyologist Hora in 1923. It contains a single genus, Cyrenacus. And when I started working on this group in 2005, there was only 6 described species. Most of my focus on this group was anatomical. I was interested in their osteology, but I also did some taxonomic projects as well. Now, many of you might be already familiar with this group, but for those of you that aren't, I just give you a little general background. So this group is distributed throughout the Indo-Burma region. It's found in foothill areas of Nepal, Bhutan, Bangladesh, and Myanmar. And it's also found, one species is found in the Western Ghats region of India. So these are rheophilic fishes. They live in fast-flowing water, and that water is usually well oxygenated and quite cool. So these are the types of habitats that you'll find Cyrenacus in. These are both in Nepal. They can be quite torrential in some places, and you can see people standing on the side of this river, just to give you an idea of scale here. In these habitats, Cyrenacus is typically found in riffles and in swifter areas, and they live on the substrate. They're benthic, and they have these very large paired fins. So here's a ventral view of a Cyrenacus in a photo aquarium. You can see the large pectoral fins here, with a very high number of unbranched rays. And if you look carefully at these rays, you can see they have this kind of thick skin that's on the ventral surface. And this skin is quite interesting. And if you look at it up close with scanning electron microscopy, what you can see is that this whitish opaque appearance is caused by this keratinized layer of skin. And the cells at the surface of this skin exhibit structures called unculi, which are keratinous structures that arise from the surface of a single epidermal cell. Unculi is plural and unculus is singular. So here's a single unculus. So these structures almost look like small hairs. What they're hypothesized to do is help fishes that have them adhere to the substrate via friction. So Cyrenacus can stick or adhere to the substrate in these fast-flowing streams using these modified cells on the ventral surface of their paired fins. They also have very interesting mouth parts. So here's an SEM of the mouth of Cyrenacus balatora. And again, what we can see here is it's kind of a field of unculi. If we look up close, this is a nice SEM because it actually shows the borders of a single epidermal cell, and each one of these cells has a single unculus. So unculi were actually described and named by Tyson Roberts in the 1980s. And they're quite diverse, and they probably have a number of different functions. So on certain parts of the body, they may be involved in adhesion, and on the mouth parts, as in the Cyrenacus, they may be involved in abrasion for scraping the substrate. So Cyrenacus has a lot of unculi on its upper lip, and it also has a very strange lower lip, which is kind of pad-like here, and it doesn't have unculi here, but what it has instead are vast fields of taste buds. And these taste buds are involved in chemosensation, of course. So Cyrenacus is a benthic fish that's feeding on the bottom, and it's probably scraping algae from the surface of, or scraping the biofilm from the surface of rocks. And whenever I get the chance, I always like to look at fishes that I study with SEM because it really gives you just a different view on these structures. And there's actually a very rich history of looking at morphology of fishes with SEM in India. And a lot of Indian scientists have studied hillstream fishes in particular, like Gyara or sisaurid catfishes with this technique. So most of my research on Cyrenacus was anatomical, but I also conducted some taxonomic work. And one taxonomic study that I conducted was focused on Cyrenacus balatora, which was described by Francis Hamilton in 1822 in his Gangetic Fishes book. This is a fish that used to be considered to have a relatively strange distribution, if you ask me. And in their summary of Indian freshwater fishes, the Telwar and Jingram described the distribution of Cyrenacus balatora as encompassing multiple drainages in multiple countries, including those of India, Bangladesh, Nepal, and also Myanmar. They also noted a lot of variation between the populations, or the so-called populations, that inhabited different drainages, including differences in stromation. And so I was interested to look at Cyrenacus balatora and assess whether this was actually a widespread species, or if it was instead a species complex of multiple species confused under one name. And I was actually able to examine material of Cyrenacus balatora from multiple areas, including both sides of the Indo-Burma ranges. And when I looked at this material in detail, what I discovered was that it wasn't just a single species. It was in fact multiple distinct species that had discrete allopatric ranges. And so I gave names to all of these species over the years. And I was able to do that because I designated a neotype for Cyrenacus balatora, which allowed me to have a comparative context. Now these fishes are pretty similar, and I did count scales and count fin rays as everyone does, but that wasn't very helpful in this case because there was almost no variation in fin ray or scale counts. And so what I discovered instead was that there was more useful variation in the color pattern of these fishes that could be used to diagnose species than there was in typical meristic counts. But it took me a long time to figure that out, and it was only after I was able to to identify the components of the color pattern, give them names, and then look for differences between the different species. So what I mean by that is that I was able to divide up the different components of the color pattern into dorsal saddles, lateral blotches, small rows of dots that were present on the scales and that were quite consistent in their location. And this is not something that, that I was the first to do. Many people have done this for different groups. For example, Fang Kuhlander did something very similar for Danio and Devario. And so this is actually very, very useful, and it gives you a way to read the diversity that exists in the color pattern and make comparisons more easily. So there's variation in body pigmentation. There's also variation in the caudal fin pigmentation as well. And all of these caudal fins here, although it looks a little bit chaotic, the patterns may seem chaotic, but once you start to look at the details, you can make sense of that chaos. And I used characters of caudal fin pigmentation also to diagnose different species. So typical meristic counts were not very useful, if you ask me, for distinguishing between species of Cyrenacus, but color pattern was actually very useful. There's also lots of variation in the skeleton of the different species of Cyrenacus, and once you prepared cleared and stain material and had the opportunity to examine that and make comparisons, some of the differences were quite dramatic and very useful for diagnosis. What I'm showing you here is some illustrations that I prepared from cleared and stained specimens. And these are simple line drawings. And some of you may have prepared line drawings like this before, but if you haven't, you might be interested in the process of how one would actually produce these line drawings. And in my case, it always starts with a photograph of a cleared and stained specimen, and this is actually quite an ugly one. I didn't clean it up intentionally to show you sometimes that what these specimens actually look like when you look at them under the scope. There's a little bit of debris here which shouldn't be there. There's a bubble here that also shouldn't be there. But I would take an image like this and I would either produce a line drawing using technical drawing pens and vellum paper. Depending on the amount of effort that you put into drawing like this, it could take anywhere from a few hours to maybe a few days, depending on how invested you are in the stippling. So this drawing in particular took me around 4 hours to produce. But in more recent years, time has become more precious, and I've actually started to cheat a little, and I no longer use pen and ink. I started to produce illustrations using digital graphics software like Adobe Illustrator, and this particular graphic actually took around 10 minutes to produce, and in my opinion it shows the features that I'm interested in, and so I think it's good enough. It's maybe not as pleasing to the eye, but it does the job. An alternative would be to take the time to clean your clear and stained specimens, and what you could do is actually produce a photograph, and that photograph could be annotated and touched up using, of course, software like Adobe Photoshop and Illustrator. And in this particular image, this is not a Cyrenacus. This is a clingfish. This particular figure took around 1 to 2 hours to produce. And the good thing about this is that you can go back and modify it, and you can add lines around the different bones to highlight certain parts. So there's many different ways to produce a scientific illustration, of course, and these are just some methods that you might want to use. But anyway, back to Cyrenacus. By studying cleared and stained material, it's very easy to see the differences that exist in the skeleton between different species, if they exist, of course. And in Cyrenacus, I noticed a number of differences that were useful for taxonomic purposes, including variation in the cephalic lateral line canals. And some members of the genus, there is a canal on the angular articular bone, and others there's not. In other species, they may have a small canal on the parietal, and others they may not. So there's variation there if you take the time to look for it. There's also a lot of variation in the fontanelle that exist on the surface of the neurocranium. And this character was actually one that I thought was very useful, and I paid a lot of attention to it. And maybe it's not so useful anymore, but when I was working on this group, it seemed to be a useful character. Of course, when we discover new species and they exhibit intermediate characteristics, a useful character can no longer be useful. There's also variation in the mouth parts of Cyrenacus, and if you look at these four SEM shots, you can see that there's pretty tremendous differences in this small flap of skin at the side of the mouth. This species here has a very rudimentary one. So there's details really everywhere if you take the time to look for them. And when I had found that variation and accumulated it, I tried to make sense of it. And what I was able to do with that was propose groupings of Cyrenacus based on this variation. And I proposed 3 species groups, the Nudifracicus species group, the balatora species group, and the Homoloptera species group. And Cyrenacus cicadio, which is the type species of the genus, I suggested didn't belong to any of these 3 groups. Subsequent phylogenetic work has corroborated these groups and suggested that they may actually be monophyletic, and so the Cyrenacus nudifracicus group and the Cyrenacus balatora group seem to be good natural groups. What's interesting about this study is that it also included the Western Ghats species Cyrenacus tenura, and based on this hypothesis, it groups with the Cyrenacus nudifracicus group, and that makes sense morphologically as well, in my opinion. So in 2005, when I started my dissertation research on Cyrenacus, there were only 6 described species. But in the last few years, the number of species within this genus is really kind of skyrocketed, in my opinion. Where even the last 2 species were described in 2020. This, might not be all the species. I'm sure there's many more to be discovered and described, but I just did not expect this diversity when I started working on the group, and I was lucky to describe a number of these new species, but but many others have actually also described a number of these new species. And of course, sometimes species are synonymized with others or treated as subspecies, and that was the case for some of the Cyrenacus as well. So Cyrenacus rowleyi was resurrected. It was previously considered to be a subspecies of Cyrenacus homoloptera, and it was elevated to full species. And then sometimes names change as well, and the name of Cyrenacus gracilis has changed to Cyrenacus nudifracicus. And all of this has happened since 2005. So there's a lot going on in the Cyrenacus world. We're now up to 31 species. Now, if we look at those species and look at the groups that they belong to, we can see that the majority belong to the Cyrenacus balatora group, and this is clearly the most diverse species group of Cyrenacus. And the Homoloptera group and the Nudifracicus group are smaller, but you know, who knows what might happen in the next few years as new species are discovered. So in my opinion, the taxonomy and classification of Cyrenacus is in pretty good shape, but not everyone agrees with me on that. And in a recent book by Heiko Blair on Indian ornamental fishes, Blair described the family Cyrenacus as a mess. He said that there was so-called homolopterids mixed in with those named Cyrenaicads. He also said that the interrelationships amongst its species are presently unknown, and he cited a study from 2007. A lot has happened since 2007, and we do have, in my opinion, a good understanding of the interrelationships of the genus. And so I don't agree with Blair. I also just want to point out that there's a lot of errors in this short chapter on Cyrenacus. For example, Blair suggests that the type species of the genus is Cyrenacus balatora. It's actually Cyrenacus sucario. And he also gives the date for Hamilton's gangetic fishes as 1922 instead of 1822. And I could go on, but I won't do that. I would encourage anyone working with Cyrenaicads not to use this as a resource. It's not the detailed account that it says on the front cover, and you would do better to work with the primary literature if you were working with this group. So to close on Cyrenacus, I just want to touch on a very interesting topic that I discovered while looking at specimens over the years, and that is sexual dimorphism in Cyrenacus. Now, what we have here is a male and female of Cyrenacus tenura, and they don't really look that different. The male has maybe some darker pigmentation, but really externally they look very similar in terms of body shape. But if you peel the skin off the surface, we start to see some radical differences between males and females. So what we have here is a male of Cyrenacus sucario and a female of Cyrenacus sucario. And if we look at the hypaxial musculature adjacent to the swim bladder, what you can see here is that it's hypertrophied in the male compared to the female. That's pretty dramatic difference between males and females, if you ask me. If we look at the structures that these muscles are attaching or associated with inside of the body, this is the fifth rib shown here in red, and also part of the Weberian apparatus shown here in blue. So these hypertrophic muscles are associated with hypertrophied parts of the skeleton, but of course only in the male. And if we look at the swim bladder, we can see that the swim bladder of the male is gigantic compared to the swim bladder of the female. The volume of the male swim bladder is anywhere between 50 to 70% larger than that of the female. And the male actually has additional chambers on the swim bladder that the female doesn't have. This is quite bizarre, if you ask me. You don't really see differences like this very often between males and females in internal organs, other than, of course, the gonads. So we can look at this in a different way. Here's a cross-section through the swim bladder and the hypertrophied hypaxial musculature. Here's a male on the left and a female on the right. This slide shows the difference in size of the swim bladder very nicely between the male and the female. And if we just schematize this so we can make sense of the details, we can see the clear difference in the size of the hypaxial musculature in the male. And there's also an additional structure here that is not present in the female. This structure is full of collagenous fibers, and it's attached to the modified hypaxial musculature, and it actually attaches to the tunica externa of the swim bladder. So there's a link between the swim bladder and this modified muscle via this strange connective tissue. So what does all this sexual dimorphism mean? Well, we really don't know, but we've made some inferences based on what we know about other fishes that have strange muscles associated with the swim bladder. And we think that what's going on here is that male Cyrenacus may be using this apparatus to make sound. Now, we have absolutely no evidence for that. We've never recorded the sound, but based on the highly modified anatomical structures here and the differences between males and females, we think that this is very likely. And it would be really great if someone would follow up on this and try to record this sound. So in my opinion, that's quite interesting and quite a major discovery for a group of fishes that were first described in the 1820s. And we've only just discovered this radical sexual dimorphism. So what that should tell you is that we can still learn new things about even relatively well-studied groups. So I want to move now from South Asia and move to Southeast Asia to talk about another group that I worked on, an interesting group of cyprinids belonging to the genus Sundadanio. These are tiny miniature cyprinids that range in size from 14 to about 18 millimeters in length, and they live in peat swamp forests in coastal areas of Sundaland, including parts of Borneo, Sumatra, and adjacent islands in between. These are incredible habitats for a number of reasons, but they have a very low pH, and I'm sure that Lucas Ruber has already told you all about these habitats, so I don't need to recover this ground. These are photographs that document differences in coloration between Sundadanio living in peat swamps in different parts of their range. And these photographs are, at least to me, suggest that this species, which is considered to be a single species may actually be another example of a species complex where multiple species may be confused under one name. So as for Cyrenacus, what I did with Sundadanio was I started to look at them in detail, and what I concluded was that this wasn't a single widespread species, but in fact it was multiple species, and almost every peat swamp that we looked at had, in my opinion, its own endemic species of Sundadanio. So again, the reason that I was able to propose multiple species here was not based on fin recounts or measurements, but it was again based on differences in the pigmentation pattern. And the pigmentation pattern in Sundadanio is not as complex as in Cyrenacus, but it has a number of components, and once you define these components and compare them between specimens, you can sometimes see quite dramatic differences. And just to give you an example, this is a structure that we referred to, or a pigmentation feature that we referred to as the dark lateral stripe. Here's the dark lateral stripe here on the side of the body. And in this particular individual, the lowermost limit of this feature actually extends ventral to the center of the eye. In this individual on the left, that's not the case. The dark lateral stripe is actually much narrower, and it doesn't extend beyond the center of the eye. So using these types of characters, we were able to diagnose multiple species. And we also noticed variation in other traits as well, including, for example, squamation. Now, we didn't count the scales in this case, but we noticed differences in the size of the scales. And we also noted differences in the extent of the scales along the caudal peduncle. So in these two upper images, and again, these are scanning electron micrographs. So this is just showing a small area of the specimen. So here's an area just behind the head. Here's the opercle. And then this is the caudal peduncle area. So the caudal fin would be here. If you compare these two upper individuals to this lower individual, you can see that these two individuals have scales on the caudal peduncle. And this lower individual only has a very small number of scales restricted to the midline of the body. And these are all adults. They're not different ontogenetic stages. And we know they're adults because they have tubercles. These are males, and tubercles are usually structures that develop in association with reproduction in cyprinids. So we're confident that the absence of scales in this lower specimen is because we just don't have them. And so the absence of scales versus the presence of scales is another example of a character that we use to diagnose Sundadanio. And this SEM here is just to show you what these tiny fishes look like up close. This is a male, this is a female, and the males have incredible tubercles all over the lower jaw, whereas the females don't. So these are the kind of details that you can get when you use techniques such as scanning electron microscopy. It really allows you to get very high resolution, high magnification information. So when we started working on Sundadanio, it was recognized as a single species, Sundadanio axelrodi, and we split it up into 8 different species, and 7 of these species were new species. And this was a study It was published in 2011 with Maurice Correlette and Yokoi Tan. And we really had fun with this project, and especially when it came to naming the different species. And if you can see some of the names here, for example, this one we called Sundadanio garguli after a gargoyle, Sundadanio echinus after a hedgehog, Sundadanio goblinus after a goblin, and Atomus in reference to its small size. So that's always a fun part of describing new species is coming up with an interesting name. Unfortunately for Sundadanio, they live in habitats that are under significant stress. These habitats are disappearing rapidly, and in many cases, the peat swamps that these fishes were collected from in the 1990s are actually no longer there, and instead, what's there instead is a palm plantation. And it's possible that some of these small fishes that were only relatively recently described may potentially already have been extirpated or even become extinct. And that might be the case for Sundadanio garguli on Banka Island. And I don't know that for a fact, but these fishes are found nowhere else, and these habitats are rapidly disappearing. And it's sad that they're disappearing because we're also robbed of an opportunity to find out more about them. And one interesting detail about Sundadanio that we discovered over the years is that there's also sexual dimorphism, internal sexual dimorphism of the hypaxial musculature. This is a male and this is a female. And what you can see here is a very large bulbous muscle, which is modified hypaxial muscle, associated with the swim bladder in the male. And the female lacks this muscle entirely, or at least it isn't developed to the same extent as it is in the male. There's also differences in the pectoral girdle, including a very large process on the clifrum in the male, which is absent in the female, and then a highly modified pectoral fin ray. And based on all of these differences, we also suspect that male Sundadanio are capable of producing sound. And this sound has actually been recorded, and aquarists have reported hearing sounds from Sundadanio for several years. So if you take the time to look at specimens, you can always find something interesting about them. And sometimes you find pretty radical differences between the sexes, which suggest that the males and females are doing something quite different. Now, whether this sound is involved in courtship or if it's associated with male-male combat, we just don't know. And these are questions that someone would need to answer through further study. So I want to move now from Southeast Asia, and I want to go to North America. This is the area that I've worked in for the last few years, and there's a very nice radiation of cyprinid fishes here in North America, and they have a long history of taxonomic study, as you would expect. There's a lot of ichthyologists in the US, and there has been for quite a long time. One of the fishes that's interested me since I've lived here in the United States is this fish here, that's Neotropis amabilis. And what I want to tell you now is kind of a tale of caution. And this tale is that you should always remember the rule of priority when you're conducting taxonomic studies. So Neotropis amabilis is commonly referred to as the Texas shiner. And if you're not familiar with Texas, this is one of the states in the southern parts of the United States, shown here in red. So the Texas shiner it's called the Texas shiner because it's found almost entirely in Texas. There are some records from Mexico and also New Mexico. But I was interested in this fish because the university that I work at is not very far away from the distribution range of this fish right here. Sorry, right here. I was interested in its phylogenetic relationships. I wanted to study it in more detail. And so we went into the field and collected specimens, and we sequenced DNA from them, and we got a result that we did not expect. And it turned out that what we considered to represent Natropus amabilis was actually two distantly related lineages of minnow. And when I say distantly related, I mean that they're separated by multiple different taxa. They're not each other's closest relatives, and they also exhibit pretty high genetic distance. This is an uncorrected p distance of 14%. So what this suggests to us is that we have two different things, and of course, we're clearly dealing with two different species. But which one of these is Natropus amabilis, and what is the other one? Is it a new species or is it a named species? So these are the questions that we had to answer. So the first thing we did, of course, was look at specimens and try to figure out if there were morphological differences between these genetically distinct entities. And it's common in the United States to follow the standards counts and measurements that were introduced by Hubbs and Logler. And so, you know, Hubbs and Logler introduced a number of measurements and counts that actually are used, you know, not just in the US, but in many regions of the world. And we collected counts and measurements on specimens of Natropus amabilis belonging to the two different lineages that I've denoted here in blue and red. And what we found was we couldn't really separate them out using these standard counts and measurements. You know, that's what we expected because these were, of course, considered to be the same species. So we didn't give up though. We kept going, and we looked a little harder at the specimens. We started to notice details that we didn't expect. Here's the lateral line canal, the root of the lateral line canal, and a member of the blue lineage and a member of the red lineage. And what you can see is that the pigment of the lateral stripe doesn't reach the lateral line canal here in this lower specimen, and it does in this upper specimen. So there's subtle differences in pigmentation in relation to the lateral line canal. We also notice subtle differences in live coloration. So this individual has a black marking on the head, which is absent in this one. Then we also noticed some internal osteological differences. So the number of differences between these two lineages started to accumulate, and so we went back to our phylogenetic data set, and we could say that we had morphological and molecular evidence to support the idea that Natropus amabilis is actually two different things. But which one is Natropus amabilis? That's another question that we got to answer. And of course, to do that, we go back to the original description. And the original description comes from Girard, a Frenchman that was working in the United States. It's a very short description, which is pretty standard for the times. And Girard didn't give many details here that we can use to, you know, they're pretty generic, but he does have something that's very important, and that's information on the type locality for the species. And the type locality is the Rio Nueces in Texas. And what we can see is that individuals from the Rio Nueces fall out within our blue clade. So that allows us to hang the name Natropus amabilis on this clade. But what about the other one? what do we do? Do we identify it as a new species? Well, sometimes we want to describe new species, but we got to be good taxonomists, and we got to make sure that what we want to describe as a new species is actually a new species. We have to check that there isn't a name available for this species. We have to check that it hasn't already been described by someone else. And it, you know, the first place that we looked is, of course, to the synonymy of Natropus amabilis. And there's actually multiple names available from the synonymy of Natropus amabilis that could apply to our red lineage. And these names actually come from Gerard, and they were actually introduced in the same publication on the same page as the name amabilis. And so we have to use one of these names to apply it to our red lineage. So which one do we pick? It could be either one. And as the first revisers, it's actually up to us to choose a name. Both of them could be applied in this case. And the name that we chose was actually Megalops. And we did that for no other reason than the fact that this fish is still present at the type locality. We didn't choose the name Socius because this fish is no longer present there. It's actually become extirpated. So we thought it would make more sense to take the name Albornus megalops. And so the take-home message here is that, you know, you should always check the literature to make sure that you're not creating a synonym. Someone may have already described the species that you're working with that you think is a new species. And of course, because of the rule of priority, the oldest name available has to be applied. And so we were able to reinstate the Natropus megalops. And we redescribed the species, and we concluded that it should be considered a priority for conservation because, unfortunately, Natropus megalops is restricted entirely to the Rio Grande drainage in Texas and Mexico. And this is actually a river that's under significant stress, and the World Wildlife Federation has named this drainage one of the world's top 10 rivers at risk, and along with some other rivers that you might be familiar with. And the problem in this area is water over extraction, but also agriculture and of course impoundment from dams. And because we recognize this fish, it now hopefully becomes on the radar of state agencies that would put measures to conserve this fish into place. And it also exhibits low levels of genetic diversity. So that's a bad thing as well. This is a haplotype network. For those of you that are unfamiliar with it, so an easy way to visualize genetic diversity, and genetic diversity is very low in this fish. If we compare it to another fish that has high genetic diversity, that's quite clear. So it's pretty common in the United States to discover that a widespread species is probably multiple species that are not currently recognized as valid. But the problem is that the work to elevate these species and recognize them usually doesn't get completed, and of course that's bad for the fishes, especially in areas where if the areas are under significant pressure from anthrogenetic factors such as urbanization and water extraction. And this is just one more example. There's many of them, and not just in the United States, but all over the world. And so we need to describe these fishes. We shouldn't let them go unrecognized. So I've spent some time talking about cyprinid fishes, maybe more time than I should have, and maybe I ask the organizers if I still have time to continue here to talk about clingfishes.
Yes, please.
It's possible.
Yes.
I'll try to be quick. I'm sorry. Some of you might not be familiar with clingfishes, and I'm really happy to get the opportunity to introduce them to you. Clingfishes belong to the family Gobiaceae. This is a predominantly marine group of percomorph fishes that are distributed globally and in temperate and tropical coastal areas, everywhere from the intertidal zone all the way down to about 500 meters. They live in coral reefs, they live in rocky shores, seagrass beds, and some of them actually inhabit fresh water, but only in the New World. What's interesting is that although this group has a global distribution, there seems to actually be kind of a dead spot on the map for these fishes. There's very few records from the Indian coast, and I would argue that they're probably there. This may be just because nobody's looking for them, and so I would be very interested to know if anyone comes across Gobiaceae in this part of the world. There are records from Sri Lanka, of course, and in parts of southern India, but I expect that there's many more clingfishes in this area that are waiting to be collected. These are the types of habitats that they occur in. They're high energy habitats, like rocky shores, lots of wave action. You can inhabit these high energy areas because of their ability to stick to the substrate. And this is a freshwater clingfish that's filmed underwater in a freshwater stream in Dominica. So they not only stick to the substrate very well, they also camouflage, match the substrate, to a very high degree also. And they really have the ability to stick to almost any substrate that they encounter in a marine environment, whether it's abiotic or biotic. They can also stick to human skin, as demonstrated here, and they also stick to things that we dump into the oceans as well. How do they stick, though? They do this with a very elaborate modification of the paired fins. So this is the so-called adhesive disc of a Gobiaceae. These adhesive discs are covered in papillae. These papillae fringe, the papillae are present all over the disc, and they have these fimbriated structures surrounding the disc. This disc is a modified pelvic fin and also contains parts of the pectoral girdle as well. So how do these stick? If we look at this papillae up close, what we see are very small micro structures that allow them to interact with the substrate. This allows them to create friction, and of course friction works well on rough surfaces, and these micro structures allow clingfishes to stick very well to rough surfaces in marine environments. And they can actually stick much better than suction cups that are manufactured, especially on these very rough surfaces. And this is a research that was conducted by Adam Summers and his group to show just how well clingfishes can suck to substrates. And I'm just going to skip this video here. But this is a workshop on taxonomy, and I just want to talk about the taxonomy of the group for a minute. The first clingfish was described in 1758, and in 2020 we're still discovering new ones. There's about 180 described species now, and there's about 50 genera. So the first species was, of course, described by Linnaeus in Systema Naturae, 1758. And we've continued to add new species of clingfishes. There's a very rapid increase in the number of described species in the 1950s, and that's the result of a study by Jack Briggs, who conducted a taxonomic revision of the group in the 50s. And this study introduced a number of new species, a number of new genera. And he also classified those taxa into subfamilies. He did that based mostly on the condition of the adhesive disc. So he recognized clingfishes that had a double disc and clingfishes that have a single disc. And then based on the configuration of the disc, he placed them into several subfamilies. But unfortunately, recent work has shown that many of these subfamilies may not be monophyletic, so they may have to be revised. In recent years, the number of newly described clingfishes are actually quite interesting. There's a number of newly discovered species that have introduced new characters to the group that weren't really known before. And these are the species that were described since 2012. There's about 20 or so species that have been described in this time period. And I just want to highlight some of these species. This is an incredible clingfish that might be the smallest species that was discovered off the Bahamas. It was described based on a single specimen, but it's been discovered to have a more wide distribution throughout this region. Another interesting species was described from Australia, and this is a species that I had the opportunity to describe. It was known from only 2 specimens, and they were collected from off the coast of Western Australia from sponge and algae reefs. 2 specimens is not a lot to go on, but we concluded that these 2 specimens actually not only represented a new species, but also represented a new genus. And the reason for that is because they have some truly bizarre characteristics of their trophic apparatus. So if we look here at the paratype, we can see here that they have this massive field of teeth in the upper jaw. And a lot of fishes have that, but that's not very common in clingfishes. And here is a CT scan of the upper and the lower jaw of this new species. And if we compare it to a regular clingfish, we can see that it has about 20 times as many teeth in the upper and the lower jaw. So this suggests that they may be feeding on something strange that other clingfishes maybe don't feed on, but of course we don't know. We only have 2 specimens. So we chose to use CT scanning instead of clearing and staining to investigate this species, because we only had 2 specimens in it, and it seemed like a bad idea to destructively sample one of them. But CT scanning is a great technique for situations like this, when you have very little material and it's precious, and you don't want to destroy it. Of course, clearing and staining is useful, and it allows us to see features that we wouldn't be able to see otherwise, but it is a destructive process, and of course there's no going back. So this was an interesting discovery, in my opinion. And we named the genus Netaramphus radula, and Netaramphus meaning duck snout, and radula after the mouth parts of a snail, which the strange jaws reminded us of. Another interesting clingfish that was only described relatively recently is Protogoby esox asymmetricus, and this was described in 2016 by Freaky et al. This is an interesting species because it is actually the deepest clingfish that's been discovered to date. It's known from down to 560 meters in depth. That's really quite deep, if you ask me. Most other clingfishes are really only known from around 5 to 20 meters, but this is kind of the lower limit so far for clingfishes. It's also interesting because the authors of this paper suggested that it was asymmetrical. Now, asymmetry is not common in nature. It exists, but prime examples of asymmetry include things like flatfishes, where the eyes are on either the left or the right side of the head. This seemed a little strange to me, so I wanted to get to the bottom of this, and I started to read the details of this paper, and it turned out that the asymmetry actually differed between individuals. So in some individuals, the asymmetry was towards the left, and others it was towards the right. This seems a little strange. So here's the holotype, and here's a drawing of the holotype. Of course, they don't really, the drawing doesn't look like the specimen, but in my opinion, it doesn't really look like it's asymmetrical. It just looks like someone has placed it down on a table and positioned it in such a way. I managed to examine the a paratype specimen of this species, and an X-ray that's available from the Paris Museum clearly shows that it's probably not asymmetrical. It's just symmetrical. Sorry, it's just asymmetrical as an artifact of preservation. And this description by Freaky et al. Of an asymmetrical clingfish, in my opinion, is actually based on a misinterpretation of anatomy. So they write that the asymmetrical bending starts after the first vertebra, but actually the bending really doesn't happen until after the 9th. So it's also an error in their paper. They also suggest that the asymmetry extends into the head, and this is only really evidenced by the illustration provided in the paper, which doesn't really provide much information. So if you ask me, I don't really buy that these fishes are asymmetrical. And also, if you ask me, I think that this fish should have been described in the genus Copua, which is another genus of deep-dwelling clingfishes that are known from Japan and Australasia. And these fishes are very similar. They have red markings on the body, which are also present in Protogoby esox. Copua has a so-called anti-tropical distribution with specimens known from Japan and Australia and New Zealand. But if we consider Protogoby esox in this context, it falls right in between those two clusters. So I would say that this fish should have been described in the genus Copua, and erecting a new genus based on asymmetry was a bad idea. And some recent genetic data suggests that Copua and Protogopa esox are actually very, very closely related. So recent years have shown us that there are still very interesting clingfishes out there, including the smallest, the deepest, and the toothiest. And I wonder what discoveries we'll find in this group in the next few years. Now, I promise this is the last little story that I'll tell you before I end today. And I know I've taken more time than I should have, but this is something that I think is very interesting, and maybe you'll also agree with me. This relates to the genus Acertus. This is a genus of clingfishes that are found in the Western Atlantic and the Caribbean region. And in 2014, Carol Baldwin, who works at the Smithsonian Institution, sent me this data set which suggested that Acertus artius was actually two different species based on genetic divergence. And she asked me if I would have a look to see if I could find any differences that would allow us to separate the two groups and potentially describe a new species. This, of course, this is quite common, and a lot of people refer to this as cryptic species these days. So Carol Baldwin sent me material, and I looked at it, and you know, there wasn't a lot of differences initially, but one feature that stood out was this opaque patch of skin just behind the opercular apparatus in the so-called deep lineage or the green lineage, which was absent in the blue lineage. And this was very interesting to me. It's not something you see every day. And I looked at it in more detail using histological sections. And what I noticed was this opaque patch of skin is actually a cluster of very large cells with very large vacuoles. These cells look very, very similar to the cells that you find inside of the pectoral, sorry, very similar to cells that you find in association with the pectoral fin spine of a catfish or the dorsal fin spine of a lionfish, for example. And these are fishes that are known to be venomous. And these cells are actually toxin-producing cells in catfishes and lionfishes. In the clingfish, these weird cells are associated with a highly modified subopercular bone, which also has a groove in it. So this groove, which is associated with these cells, which look like toxin-producing cells based on their appearance, might suggest that this small clingfish may be toxic. So the first thing that we, of course, had to do was figure out which of these two species was Acertus artius, and we did that by examining the type material, and when we examined the type material, we also noticed this feature, and so we could name, or we could place the name Acertus artius with this form, and we concluded that this other form, as Carol initially suspected, was a new species or a cryptic species. And so, although these fishes look superficially similar, they have very different traits. And this combination of traits suggests to us that Acertus artius may actually be a venomous clingfish. And we propose that the modified cells may be producing some type of toxic substance, which is delivered via this very sharp spine-like process on the subopercle. And we haven't followed up with this, and we have no evidence to support that these cells produce toxin, but based on their similarity to other cells and the toxin glands of other fishes, we think that this is quite possible. So what we have here is potentially a toxic clingfish, and of course there's other groups of toxic fishes out there, but clingfishes were not really considered to be venomous before, and we think we might have the first example. So what's truly remarkable, and this is the reason I wanted to tell you about this, is that we found something very similar in Arcos nudus. And Arcos nudus, of course, was the first clingfish to ever be described, and that was by Linnaeus in 1758. When we look at Arcos nudus, we see the same modifications. We see a spine-like subopercular bone that's associated with a gland that contains these very large cells with a very large vacuole. And this cluster of cells is again associated with a very sharp spine-like subopercular bone. So almost 250 years after Linnaeus described this fish, we're still learning something new about it. And the take-home message here is that you should really take the time to look at fishes in detail when you have the chance, because when you pay attention to these little details, you sometimes find interesting characters that can lead to interesting discoveries. So thanks for your time. I'd like to say thanks to my collaborators and grad students and funding agencies that have allowed me to do some of this research.